Hello, in this video I'll be going over the development of the skeleton. We'll begin our journey at the end of the 4th week of gestation. What I have drawn here is a transverse section of an embryo. This will be the dorsal part of the embryo or the back. This is the ventral part or the belly side, and then these are the lateral parts of the embryo. In blue, so we'll just go over those germ layers real quick as a refresher. Blue is the ectoderm. This part of the ectoderm will form the skin. This part of the ectoderm is called the neural tube. It will form the spinal cord and the brain. The other germ layer is the mesoderm in red. There's different parts of the mesoderm we'll go over in just a second. And then in yellow, right here, is called the endoderm that will form the future gut tube. Now, we're talking about bones in this lecture. There's going to be different germ layers that will contribute to the formation of the skeleton. First, I wanted to highlight this part of the mesoderm here. This part of the mesoderm is called paraxial mesoderm. And the paraxial mesoderm will form somites or little balls all along the length of the back of the embryo. And those little balls will further differentiate into what's called the sclerotome, the dermatome, myotome. For our purposes, we're interested in the sclerotome. But in general, the paraxial mesoderm is going to form the vertebra and the ribs, as well as the skull. And then the other germ layer that we're interested in for this video is called the lateral plate mesoderm. So this is part of the lateral plate mesoderm here. This is the visceral layer, and then we have the parietal layer of the lateral plate mesoderm. So it is this lateral plate mesoderm, specifically the parietal layer. You might have also heard it referred to as somatic. And the parietal layer of the lateral plate mesoderm is going to form the bones of the appendicular skeleton, or the bones of the limbs, and the sternum. And then finally, remember during neurulation, or the formation of the neural tube, there are going to be what are called neural crest cells that are going to form kind of on that dorsal margin of the neural tube. And they're going to migrate and they're going to form different things. In terms of the skeleton, the neural crest cells are going to form the skull. Now that we've talked about the different germ layers that are going to contribute to the formation of the bones, we're going to talk about two different ways that bone is going to form. And those two different ways are intramembranous ossification and endochondral ossification. So, actually, I wanted to mention this real quick. The neural crest cells, paraxial mesoderm, and lateral plate mesoderm, collectively, as these embryonic tissues distribute and go to different parts of the body, they're going to form kind of a loosely organized embryonic matrix, and collectively that is called mesenchyme. So when you see the word mesenchyme, it could include neural crest cells, paraxial mesoderm, lateral plate mesoderm, a bunch of different things. So in terms of intramembranous ossification, you're going to have mesenchymal cells that are going to directly ossify and form osteocytes. And then for endochondral ossification, it's going to be a very different process. We have mesenchymal cells, and mesenchymal cells are going to differentiate into chondrocytes, and then the chondrocytes are going to differentiate into osteocytes. So intramembranous ossification, we go from a neural crest cell, let's say, mesenchyme, directly to osteocytes, and in endochondral ossification, mesenchyme forms chondrocytes, which will foreshadow the future bone, and it will eventually turn into osteocytes. All right, so now we're going to shift gears and talk about the axial skeleton, specifically the skull. The skull is subdivided into the neurocranium, or the brain case, So this would be that neurocranium right here. And the neurocranium is further subdivided into the membranous part. This would be the calvarium right here, made up those flat bones that make up that skull cap. And then there's also the cartilaginous part, which is drawn out here on the right. If you could imagine, if I were to remove the calvarium like this and take a look on the inside, this is what I would see. That'd be that skull base. That'd be the cartilaginous part. And then finally, there is the viscerocranium, and this is generally where that viscerocranium would be located. Those make up the bones of the face. The skull itself will actually undergo both intramembranous and endochondral ossification. And if you noticed, I actually color-coded this in such a way down here to show you what parts actually undergo intramembranous and endochondral ossification. So the neurocranium, the reason why it's split up into two different parts is they actually develop differently. The membranous part will develop via intramembranous ossification, so mesenchymal cells directly to bone, and the cartilaginous part, the base of the skull, will go from mesenchyme to chondrocytes, and then the osteoblasts will migrate in to form the final bony product. But specifically, the mesenchyme is going to be composed of different germ layers. So what I mean by that is there are going to be neural crest cells and paraxial mesoderm that are going to be forming the skull itself. So now let's get into that. Looking at the membranous part, the parts that are going to be derived from neural crest cells is the frontal bone. And here's that frontal bone right here. And then the parts that are going to be derived from paraxial mesoderm are the squamous part of the occipital bone and the parietal bone. So here's that parietal bone and then the occipital bone right there. The cartilaginous part, or the base of the skull, is going to be derived from specifically the anterior part. So we have the sella turcica right here in yellow. This is that sella turcica. Anterior to the sella turcica, so this is anterior and this is posterior. Anterior to the sella turcica, these bones are going to be derived from neural crest cells. That will include the sphenoid bone and the ethmoid. Those are going to be derived from neural crest cells. And then posterior to the sella turcica, this region right here is all going to be derived from paraxial mesoderm. This will be specifically the mastoid and petrous parts of the temporal bone, and then the base of the occipital bone, and then finally, if you notice here, the sphenoid anteriorly is neural crest cells, and posteriorly it will be derived from paraxial mesoderm. So here is that petrous part of the temporal bone. Here's part of that sphenoid bone here, and then the temporal part of the squamous part of the temporal bone is going to be derived from neural crest cells. So now moving on to our viscerocranium. The viscerocranium, it's going to form via intramembranous ossification. Thankfully, all of these bones are going to be derived via neural crest cells. All we have to do is just list them out. So here's our viscerocranium. That will include the squamous part of the temporal bone. That will include the mandible and the maxilla. It will include the lacrimal bones. Oh no, there we go. The nasal bones, vomer. I think you get the idea. The only one that doesn't quite fit into this mold is the inferior nasal concha bone. That's going to be derived via endochondral ossification, but that's pretty granular detail. So that's pretty much how the skull itself is going to form. The face itself is actually going to look pretty babyish. at the start, and that's due to the absence of the paranasal sinuses like the maxillary sinus, phrenoid sinus, and you also have the absence of teeth, and as those develop, it starts to look more like an adult. The skull itself has unossified regions. Let me get a different color here. Have these unossified regions called fontanelles. And the function of these fontanelles, it's important that they remain unossified, is it allows flexibility of the skull during childbirth. And this baby's head is going to get a lot bigger, so it allows expansion of the skull as the brain enlarges during early childhood. Each of these fontanelles have different names, and I'll let you look that one up. But slowly over time, about 2 years of age, these fontanelles will have closed. There are also sutures in between each of these bones. These sutures are flexible fibrous joints. That also allows for the head to compress and pass through the birth canal, and also allows for that brain growth as well. The sutures gradually fuse over time. They take a lot longer to ossify compared to the fontanelles. In fact, some of these sutures remain open until well into adulthood. So that is how the skull will form. The other component of the axial skeleton is the vertebral column. So we'll now move into talking about the vertebral column. And we'll zoom in here on our transverse section of the embryo. We have the vertebra themselves are going to be derived from the paraxial mesoderm. So this here, ooh, let me turn my pen on. This here is our paraxial mesoderm, and the paraxial mesoderm is actually going to differentiate. Because why not? It's going to differentiate into our sclerotome. It's also going to differentiate into, losing my voice here, into the dermatomyotome. And the dermatomyotome is going to be forming the dermis and the muscles. But we are mainly concerned with the sclerotome here and how it's going to contribute to the formation of the vertebral column. So here in blue, we have the neural tube. Neural tube is going to form the spinal cord. And then right here in purple is the notochord. And they're both going to be very integral to the development of the vertebral column. So the sclerotome is going to essentially grow around these structures, around the neural tube and around the notochord. And I will demonstrate that here. So I drew out the notochord right here in purple. It's a tube, so we're looking at the longitudinal section here. And these balls here are, let's do this color, the paraxial mesoderm, specifically those somites, now differentiated into sclerotomes. So these are our sclerotome balls. And I labeled them C1, so this is C1, we got C2, C3, and C4. Interestingly enough, each of these balls are going to resegment. They're going to detach right at the middle, approximately, and join up with their neighbor. So here, C2 is going to join with C3. This part here, C3, is going to join up with C4. Simultaneously, they're going to be migrating around this notochord here, just like so. So this is what that looks like. Let's see if I can get both in the image. There we go. This is our resegmented sclerotome, forming those vertebral bodies. These are our vertebral bodies. If you can imagine, let's say we're looking at this one here. These two came together. So we have this part of C2 and this part of C3. This might be C3 and a little bit of C4 and so on and so forth. I think you get the idea. The notochord itself is going to largely regress, and it's going to form the jelly-like center of the joints between the vertebral bodies called the nucleus pulposus. The annulus fibrosus, the tougher connective tissue part is going to be formed by the sclerotome itself. So that's how the development of the vertebral cord, that's what it looks like. So what I have drawn here on the left are those vertebral bodies. I don't have them color coded anymore, but we have these vertebral bodies, and then here is the nucleus pulposus, remnant of the notochord. Remember, the vertebral bodies are going to be located right here. There's going to be a part that's going to be growing off of it called the neural arch, and that neural arch will be surrounding our neural tube. So this is our neural tube. Coming off of the vertebral bodies themselves are going to be projections. We have the spinous process coming off that neural arch and the transverse processes. Here's a spinous process, transverse process. Coming off of the vertebra itself is going to be the ribs. The ribs, derived from sclerotome, are going to extend laterally from the developing vertebra bones. And I don't know if I had mentioned this, but the vertebra and the ribs are going to be developing via endochondral ossification. So right now, they're just cartilage, but eventually they will then ossify. And this ossification begins around week 7, so pretty quick actually. The ribs themselves, as they're extending laterally, as they move anteriorly towards the sternum bone, I don't know if you remember adult anatomy, but the anteriormost part is cartilaginous in adults. So that part actually does not undergo ossification. It remains cartilaginous, but the rest of the bone will then ossify via endochondral ossification. One more bone we need to talk about for the axial skeleton, and that is the sternum bone. It's going to be derived from lateral plate mesoderm, specifically the parietal layer. So lateral plate mesoderm, specifically the parietal layer or somatic layer. The sternum will actually be paired hyaline. It'll undergo endochondral ossification as well, so cartilaginous precursor. It will be a paired hyaline cartilage models or sternal bars. Here are sternal bars. This is our manubrium. These sternal bars are going to migrate medially, and eventually they will then fuse and then ossify to form the sternum bone. So here's our manubrium, sternum bone, and then the xiphoid. The xiphoid, interestingly enough, it actually ossifies a lot later than life compared to the rest of the sternum. And that is pretty much how the axial skeleton develops. And now we'll move on to discussing how the appendicular skeleton develops. The limbs are going to develop via endochondral ossification, and the mesenchyme that are going to form the bones and the connective tissue are the lateral plate mesoderm, specifically that parietal layer. So we'll zoom in here onto our transverse section. I have a whole video that goes into limb bud development, or the development of the limbs. So this is going to be a quick and dirty version. So I recommend, if you want to learn more about limb development, watching that video. But essentially, the ectoderm is going to thicken on the lateral body wall. That thickening is called the apical ectodermal ridge, or AER. And that thickening is going to drive the elongation of this limb. And the limb is going to have a hand paddle, which will eventually form the fingers and toes when apoptosis occurs. So here are fingers and toes. The lateral plate mesoderm is going to migrate into the developing limb bud, and as the AER moves distally and further away from the mesenchyme, that mesenchyme is going to differentiate into the cartilage models. So we'll do the upper limb. Here is the humerus. Here's the radius, ulna, we have some carpal bones, and then the actual digital rays. So here are those cartilaginous models. While the cartilaginous models are being formed, synovial joints are going to form between those developing cartilages, and those areas between the contrifying bone are called interzones. We have our cartilaginous model here in our limbs. The ossification centers form first at the diaphysis. The diaphysis is the shaft of the bone, of most long bones. This occurs at around week 12. And this ossification progresses all the way to almost to the ends of the cartilaginous model. At birth, the diaphysis is completely ossified, whereas these ends here are still cartilaginous. In fact, if you look at an x-ray of an infant, you'll notice that you can't see the carpal bones because they're still cartilage, or you won't be able to see a very well-defined ends of these bones. Shortly after birth, the ends of these bones, so the ends are called the epiphyses, they will then begin to ossify. Because baby is soon going to be up and walking. So the epiphyses will ossify, but a layer of cartilage is going to remain, and this layer of cartilage is known as the epiphyseal plate. I am tired of the color pink. We are going to do green. So here is our epiphyseal plate, and this is going to function in allowing or continuing the growth and proliferation of bone and allowing the lengthening of that bone until skeletal maturity is complete. This is about age 14 to 19 years. And then at that point, it will then fully ossify. All right, and that pretty much wraps up this video. I hope this helps, and happy studying.